Thanks very much. Thank you so much for the organizers for the invitation. This has been wonderful. Very impressed with the research setup you guys have here and the quality of the science, and particularly the quality of the trainees. I was originally going to talk, kind of gear my talk more towards something more clinically relevant, more of a developed story, but I was inspired by kind of the fundamental science happening at this conference, and wanted to talk about something I'm very excited about. But the data is very raw, and as you'll see, my thinking around it is very raw, and so it really appreciate feedback and advice as I go through this. So I always like to start with the motivation. Why am I interested in the human microbiome, or say generally the mammalian microbiome? What does the microbiome do? Ultimately, we understand this, just like anything in biology, by breaking the system, raising mice without a microbiome in the germ-free setting. And then with these mice, and a lot of my research is focused on diabetes, we see all sorts of very peculiar phenotypes. As one example, the germ-free mouse is a smaller pancreas, and the beta cells secrete less insulin. The germ-free mice are more susceptible to developing type 1 diabetes in the genetic model of type 1 diabetes, and they develop more rapid onset of the disease. They have a lower body temperature, and this is, there's been some nice, elegant mechanistic work showing that this is related to brown fat and microbiome having some control over brown fat. There's impaired nutrient absorption. Some of the most obvious phenotypes are really behavioral and neurodevelopmental. And anybody who's worked with germ-free mice can attest to this. And then even all sorts of wild things. Something I'm not going to talk about. Part of my lab study is the role of the microbiome in exercise, and some beautiful work that started with Christoph Theis at UPenn, showing that not only do germ-free mice exercise less voluntarily, but they lack the motivation to exercise, and this has been tied to this really neat mechanism of enteric nervous system talking to the central nervous system and dopamine signaling in the brain. And then what's really remarkable, and why I think it's so important in medicine, is then you take a conventional microbiome, a normal microbiome, and you give it to these germ-free mice, and depending on the developmental time window, you can rescue these phenotypes to some extent, and some phenotypes stronger than others. But what it shows is, within the microbiome, if we understand the system better, this is a powerful new source of therapies for many diseases. So what I want to talk about particularly today is a question that has been on my mind for a long time, and I think a lot of people in the field. The microbiome appears to cooperate and provide benefit to its host. As you can see from all these examples, it appears that there's some dependency on the mammalian, on the mammal, on its microbiome, and then the big question is why? Why would this be? We've seen some very beautiful talks in ecology and evolution. This is usually an introduction that I use for microbiology departments in medical schools, where of course the important thing to understand is in microbiology in medical schools, the focus, is very much on pathogenesis and microbes as pathogens, with a long history, right, going back to Pasteur, and this idea of a potentially mutualistic microbiome. It's a new concept, and it's not nearly as well studied as it is in other systems, as in invertebrates. And I couldn't point to a single example of a mutualist in the mammalian gut microbiome. And I would be interested if anyone here could. And the reason is not because they don't exist. Just the work hasn't been done. I think ultimately. So then there's a question of coevolution. So despite the fact that coevolution is used in the introduction to many microbiome papers in the human microbiome, in the context of the human microbiome, mammalian microbiome, there is not evidence of coevolution to date, and the main reason is because there isn't evidence of faithful transmission across generations. And Emily mentioned the overall low heritability of the microbiome. And so vertical transmission is not observed in mammals. And so this is something that I can attest to, having looked at these kinds of data sets of mothers, the mother microbiome, and the baby, and then looking for strain sharing. And it's surprisingly small, very, very small number of strains that are shared, particularly after 3 years of age. So this is critical, of course, for coevolution to develop, but we don't see it. Just kind of from another review, the importance of parent-offspring transmission is unclear. And so this is kind of what I want to talk about. I want to take a step back and explain how I got into this. Ultimately, I was originally interested in, and still am, in autoimmune diseases, and trying to understand why there's a rapid rise in autoimmune diseases. We've, in the past many decades, gotten really good control, just really amazing work that's been done in microbiology to rein in infectious disease and this a terrible burden that historically it's had on human life. In the same time period, we see the increases in these immune-mediated diseases, particularly autoimmune diseases and allergies. And so this correlation, is there some causal element here? We can look at it geographically as well. Generally speaking, childhood diarrheal diseases are most common in lower GDP countries, generally speaking, and there are exceptions. And then we look at the incidence of type 1 diabetes, and it's pretty much the mirror opposite. And so, this correlation, again, is there some causal component? Could these microbes that cause disease in early life potentially be protective? So this is work that I did a while ago. I'm not going to go into details of this work, except to kind of set the stage, where we were studying the gut microbiome in infants from birth until they're 3 years old in Helsinki, where there's the highest incidence of type 1 diabetes in the world, and comparing them to infants across the border in Russian Karelia, where this is a border that's moved back and forth with the wars, and so genetically they're very similar. even culturally. The big difference, Helsinki, very generally urban, wealthy, and Russian Karelia, more rural, kind of traditional lifestyle. And so, long story short, what we found, particularly in the first year of life, is that in this more rural microbiome, counterintuitively, a more inflammatory kind of a microbiome, which we think is important for immune education. And particularly, it was subtypes of LPS that are present in those microbes. Whereas in the urban environment, by comparison, much less inflammatory. This got me thinking, is there something maybe that's worth studying in these rural microbiomes that could tell us some kind of a protective factor in autoimmunity, potentially. This got me thinking, well, why not go further back in time? I think ultimately I'm interested in understanding the origins of the microbiome, and this got me to think about studying foragers or hunter-gatherers. And I think it's important to say that this is ultimately the lifestyle that we've evolved under, right, for 50,000 generations, and agriculture and industrialization has happened kind of in the blink of an eye, so I think in medicine we have much to learn. There's this concept of a mismatch hypothesis between the lifestyle of what we're evolved for and the lifestyle that we live today, and this being a contributor to chronic diseases, right? So there's much to be learned, I think, from studying hunter-gatherers. We talk about health. The age of death for Americans is 7 to 8 years old. And for hunter-gatherers, I mean, it is true that the average is much lower. But that's often, people will say, oh, they don't live to old age. They do live to old age. It's just a bimodal distribution. And the reason is infection in early life is common, death in early life, and also death during childbirth. But for people who make it past those milestones, they do live to old age, and actually not too much younger than Americans. Of course, in the U.S. and most of this death is chronic disease, the major cause. For hunter-gatherers, we don't, of course, there are no hospitals, right, for official diagnoses, but generally this is thought to be caused by infections and chronic disease, very, very rare. I'm just going to grab a drink. And there is some data here. This is a study of the Tsimane hunter-gatherers in the Bolivian part of the Amazon, they're compared to a bunch of Westerners. And the amazing thing is somehow they got 700 of the Tsimane into a CT scanner in order to measure coronary artery calcium, which is a strong predictor of future heart disease. And I'm going to show you some scores, and just for context, anything above 100 is moderate artery cloggage, and anything over 400 is really extensive. And so might be a little bit tough to see, but for white males, the 50th percentile, by the time they're 75 to 84 years old, the score is 400, and so this is thought to be a normal aspect of human aging. But in the Simony men, in the same age group, the 50th percentile, the reading is 0, and you have to go to the 95th percentile, and even there, it's below 400. So I think really challenging the notion of what we think of as normal aging. So what about the microbiome? There isn't direct evidence in humans, but there's some really great work that's been done in mice, studying wild field mice, and in transplanting, doing a fecal microbiota transplant into laboratory mice, germ-free laboratory mice, and studying the phenotypes. And so just really crazy things where they challenged the mice with influenza and look at survival after infection. And so the mice that receive a wild microbiome are highly protected, whereas those with the lab microbiome are not. And then they induced colon cancer in these mice, and again find that the mice with the wild microbiome are protected. Recently, they did a study, this all from Barbara Rearman's lab at NIH, showing that the wild microbiome can protect against diet-induced obesity. And the phenotype is really dramatic, where this is a lab mouse on a high-fat diet, and then down here is the wild mouse. So same genetics, right? Just the wild microbiome, but under the same high-fat diet, completely protected against weight gain and also better insulin sensitivity. So I was motivated to understand not just in modern hunter-gatherers, because there aren't so many left, unfortunately. And those that are left all do have some form of contact with the industrialized world. By coincidence, I started working with archaeologists who happen to have plenty of, in these dig sites, particularly in the southwestern US, access to what they call coprolites or paleofeces, which is really just ancient human poop. And so some of these specimens, they're generally 1,000 to 2,000 years old that we'd studied. But the DNA, the quality of the DNA, of course, damaged, but still good enough to be able to reconstruct genomes. And demonstrate a concept that had been brewing in the field, this idea of extinctions that are happening in the microbiome. And here, we see many genomes that are shared in these different geographic distances that we don't see today. But I think what really struck me was doing time-measured phylogenetic analysis, where we're building phylogenetic trees of one microbe at a time. And this is a tree for Methanobreviobacter smithii. So these are all isolates or genomes from industrialized people or modern people, and these two are from different regions of ancient North America, and we're interested in the common ancestor, and the common ancestor existed 33,000 years ago, which is right around the time that humans were crossing into first populating America through the Bering Strait, and so this to me was really peculiar because it does suggest that there may be some transgenerational, faithful, uninterrupted transgenerational transmission of microbes over time. And it may not be vertical in the strict sense of mother to child. So some of the more recent work that we're doing is studying the Yanomami, a tribe in the Amazon, and a really wild story for how we got access to those samples. We're working with Emma Allen Vercoe in Guelph, who's just an incredible microbiologist. And so what I want to show you here is a beta diversity plot where all of the circles are hunter-gatherers. And all of these industrial microbes, these are all individuals from the industrialized world, USA, Sweden, China. And you know, what's really remarkable is how similar all the industrialized people are to each other, regardless of the geography. And then here we have the hunter-gatherers. Most of these samples are from really remarkable work that's done by Justin Sonnenberg at Stanford of the Hadza who live in Africa. And then what you see when we add in these samples, the ancient North Americans and the Amazonians, that they overlay really nicely with the Hadza from Africa, despite the fact that these are completely different time periods, completely different continents even. So what could explain this? And this is a look at all of the species that are present across all of these same samples, where each row here is a unique species. Obviously far too many to list. And all the hunter-gatherers are to the left side and industrial to the right. These are Africa, most of the samples, and then the Amazonian and the ancient. And then what really jumps out, obviously, is again, species composition, just how different these are. And most of, we study the human microbiome, we're studying this. This is our focus. But what about all this, right? These don't even have names. They're all just numbers, right? The genus, species name, and even genus names. So one thing, and I'll get back to the to the original question, and I'll wrap up. Is the population size that I think is really critical here. So this is a picture of the Yanomami, and they live in these relatively small tribes, maybe 40, 100 people, and they're nomadic, and they move around the Amazon. And this is in stark contrast to the modern industrialized world, which is highly interconnected. And it's important to note, generally microbes composition comes from exposure to other people. Also important to note that generally human gut microbes, what we detect there, they're not environmental bacteria. Some are, some are, but generally these are strict anaerobes that thrive in the human gut, or at least the mammalian gut. And so generally for us, they get transmitted through social contacts, and there's been some really great work describing how that happens. So why is all of this important? Getting back to this question, why does the microbiome appear to cooperate and provide benefit? obviously, I don't know. But here's how I think about it. If you're a microbe in a hunter-gatherer, and you know, the hunter, you know, you're in a small tribe, 50, 100 people, and you might, maybe there's 400 people possibly you ever have contact with. That microbe, what kind of a relationship is it going to have with the host? What's a good survival relationship in the spectrum of symbiosis, parasitism to mutualism? Does it make sense to be a pathogen in this context? I would say whatever the microbe can do to keep its host alive is ultimately going to benefit the microbe and be to its own advantage. But this becomes very different in the industrialized world. Because the microbe has many options, it's no longer tied to just a single individual and its relatives, but really, you know, all of their social contacts. So then the microbe, it doesn't have that same selective pressure to influence the fitness of its host. And so then from this, if there is something like transgenerational transmission, faithful transmission, then maybe we can get a clear sense of how mutualism may emerge in the mammalian gut. So let's say there's a human gut microbe. It has a new mutation. It can degrade fiber more efficiently. That's good for the microbe. As a result, it ferments this fiber into short-chain fatty acids. As we're understanding from the literature, the short-chain fatty acids have all sorts of positive health benefits for the host. So if this increases the fitness of the host, then ultimately this will increase the fitness of the microbe. Let's imagine a different mutation, and now the microbe is able to degrade mucin and derive energy from the mucus layer of the gut. That's good for the microbe, it gets this extra energy. But if this results in decreased barrier integrity, it really starts degrading the mucus layer, and this results in negative fitness for the host. Ultimately, this is going to be detrimental to the microbe. So this is a hypothesis still in progress, and data that we're still developing and working on. But I'd like to thank the many people who were involved in this work over the years, and thank you so much for your attention.